Happy day everyone. Today in this video we will continue the biomolecules chapter. We have discussed about what is bio and what is amino acid and the structure of it, and as well as we discussed about lipids. Now we will talk about nucleic acid. Nucleic acid nothing but DNA and RNA in our living system, as you all know, and these are the genetic material. Now we will see how this nucleic acid is formed. This nucleic acid is formed by the addition of nucleotides. Okay, nucleotides. Whenever we say nucleic acid is formed by the addition of nucleotides. Now we will talk about the nucleotides in detail. What are the materials which are formed the nucleotides? The nucleotides are made up of three materials basically. One is heterocyclic compounds, which is also called as nitrogenous bases. Another is monosaccharide and another is phosphate. Now, if we talk about the nitrogenous bases, it is again classified into two types, which are nothing but the pyrimidine and purine. Okay, within pyrimidine we have three, which are thymine, cytosine and uracil. In case of purine, we have two nitrogenous bases, which are adenine and guanine. Okay, in case of DNA, we will be having thymine, cytosine, adenine and guanine. So now in case of RNA, there will be a difference. So instead of thymine, we will be having uracil in case of RNA. Okay, so if DNA is present, thymine will be there. If RNA is the genetic material, then uracil will be there. Now we will talk about the monosaccharide. Now in case of RNA, we will be having the monosaccharide as ribose. This is the structure of ribose. Now in case of DNA, we will have deoxyribose, right? So what is the deoxyribose? If you see in the second place, we have OH here. In case of ribose, which is for RNA. But in case of DNA, the deoxyribose won't be having O, that is oxygen. Now you are getting the name right. Deoxy means there is no oxygen. So in the structure, in the second position, O will be removed in case of deoxyribose. So the monosaccharide will be ribose in case of RNA and deoxyribose in case of DNA, and a phosphate group will be present. These are the combination for the nucleotide: nitrogenous bases, monosaccharide and phosphate. Now, what is nucleoside? Nucleoside is nothing but the nitrogenous bases plus monosaccharide is called as the nucleoside. Okay, so the difference between the nucleotide and nucleoside will be the phosphate group. Whenever we say nucleoside, nitrogenous bases plus monosaccharide will have the nucleosides. If we add phosphate to it, it will become the nucleotide, and these nucleotide only form the nucleic acid. You are clear? And whenever we talk about nucleoside, it will be called as adenosine, cytidine. But whenever we say as nucleotides, we will call it as adenocyclic acid, cytocyclic acid. Now we are getting the difference, no? This is how the formation of nucleic acid is happening. Now we will discuss the questions from the latest topics we discussed. So the first question will be, what is the size of the lipid? Try to recall or revise it once and answer. It's 800 Dalton. Lipid found in dash fraction, that is acid soluble or acid insoluble? Acid insoluble. How many carbons are found in palmitic acid? 16, right? Low melting point. What are the lipids which are having the low melting point? Fats or oils? Oils. Lecithin is dash. It's a phospholipid. Uracil is found in DNA or RNA? Uracil is found in RNA. In case of DNA, it will be thymine. Adenosine is a nucleotide or nucleoside? It's a nucleoside. I hope you should have been answered to all the questions. If not, no problem. Just revise the notes which you have taken from the video and try to answer the question. We will see in the next topic.